Hello everyone, I am Akhil. My other group members are Mrunal, Gargi, and Neha. We are here to present a research paper on metabolic flux redirection from a central metabolic pathway towards a synthetic pathway using a metabolic toggle switch. In this research paper, scientists of Japan have used the concept of metabolic toggle switch in order to increase the yield and titer of the desired product, that is of isopropanol. Before going into deep about this research paper, let's talk about the other ways that can be used to increase the yield and titer of the product. It can be done by overexpression of target gene, deletion of target gene, or by using synthetic genetic circuits that can imitate natural gene expression. There are many problems associated with deletion and overexpression of target genes. Overexpression and deletion of desired gene may increase the titer and yield of desired product but can affect the cell growth and maintenance and perhaps results in cell death. Let's find out the basic reason behind it. This is possible because one gene can have multiple functions. For example, it can help in increasing the yield of the product, but on the other hand, it may prohibit the cell growth and maintenance. For example, in case of E. coli, glucose and lactose along with citrate synthase gene helps in growth and maintenance of cell. But citrate synthase, on the other hand, prohibit the production of secondary metabolites such as isopropanol, which is our desired product. So, in order to deal with such problem, the third approach is to use synthetic genetic circuits that imitate natural gene expression system. For example, we can use the synthetic compound that would mimic the natural available compound in function, but it would be used for a specific time interval as a switch which would be on and off depending on regulated gene expression. In this research paper, they have used the concept of genetic circuit or metabolic toggle switch in order to increase the productivity of desired product. It is an alternative approach in which we can keep the expression of particular gene high until an adequate cell mass is achieved, then turn this gene off. For example, instead of permanently knocking out a gene from the chromosome, a novel gene expression control system would conditionally inhibit the expression of a specific gene. By using this method, we can solve the problem of deleting or overexpressing the target gene for increasing the yield. Let's find out how they have done it. Their main focus was citrate synthase, which is an enzyme that catalyzes the condensation reaction of one molecule of acetyl coenzyme and one molecule of oxaloacetate to one molecule of acetate in TCA cycle. Its main function is to promote the cell growth and maintenance, but on the other hand, citrate synthase prohibit the production of isopropanol, which is their desired product. So what they did is they target the citrate synthase gene, that is GLTA gene. They induce the expression of this gene for a specific time interval so that proper cell growth and maintenance could take place. But after that, they switch off the gene so that production of isopropanol could take place because citrate synthase was inhibiting the production of isopropanol. For doing this, they used IPGT, which is a compound similar to lactose, whose function is to inhibit the expression of citrate synthase gene. The E. coli would sustain this IPGT because it is mimicking the lactose, which helps in cell growth and maintenance. Now in lab, they use different strains of E. coli and different plasmid vector to get their desired result. Now I am handing over this research paper to Gargi. She will explain about the material and method used by them.
Thank you, Akhil, for the detailed introduction. Now, moving forward to the materials and methods. So, first of all we need to do is to design the synthetic metabolic pathway for the production of isopropanol. And here the metabolic intermediate is used as the precursor that can be constructed in a host microorganisms. In this study, they have taken E. coli as their host microorganism. Commercially available chemicals and reagents are used here. For the plasmid preparation, E. coli strains were cultured in LB medium at 37 degrees and then the 1% of it were inoculated into M9 minimal medium. Strains producing isopropanol were inoculated in slightly higher glucose concentration than the non-producing one, and the final plasmid produced were by multi-step processes by additions and deletions, and the addition and the introducing different strains, different plasmids in different strains. The final plasmid produced were PTA 669, which is a switch plasmid, PTA 216 repressor plasmid, and PTA 147 plasmid responsible for isopropanol production pathway. By introducing the plasmid PTA 669 in strain TA 1334, a new strain TA 1357 was obtained, and by introducing the plasmid TA 659 into the strain TA1041, TA1414 strain was obtained. Then in these two strains, that is TA1414 and TA357, the plasmid PTA147 was introduced, which contains the isopropanol production pathway genes, producing the strains TA1415 and TA1424. The strains with the GLTA knockout genes were also produced, which are TA116, TA1184 and JW0710. Then the citrate synthase assay was carried out for the determination of activity of citrate synthase, that is the gene product of GLTA, which is done by the commercially available kits. And then for the measurement of extracellular metabolites, OD was taken, and for its quantification, chromatographic techniques were used. And lastly, they measured intracellular metabolite by using various techniques. And for the results of it, I will hand over to Neha. Thank you.
Thank you, Gargi, for the detailed description of materials and methods. And here are the results. First, we see the construction of genetic toggle switch and the regulation of GLTA expression. Here, the first figure demonstrates the design of metabolic toggle switch, and it is divided into three parts. The first is the repressor source, that is PTA216. Second is switching plasmid, that is PTA669. And the third is isopropanol pathway, that is PTA147. In this, the switching GLTA off is caused by the repression of GLTA transcription from the PTET-O1 promoter by the TET-R repressor. This TET-R repressor is encoded by the TET-R under the PLAC-O1 promoter, and its expression is induced by the addition of 0.1 millimolar IPTG. Coming to the second figure, it shows the profiles of the strains used. In this, in the absence of IPTG, the citrate synthase activity of TA1357 containing a GLTA off switch on the medium copy plasmid was 2.8 times higher than that of the control strain TA1414. in which the citrate synthase is expressed from the native chromosomal GLTA. Now, this graph represents the conditional citrate synthase activity regulation by switching GLTA off. Here, when 0.1 millimolar IPTG was added to TA1357 after 9 hours of cultivation, its CS activity decreased by 93% within 4 hours. This all indicates that in the presence of IPTG, the specific growth rate of TA1357 at logarithmic growth rate decreased by about 67% compared to TA1414 and TA1357 without IPTG. And in the absence of IPTG, the CS activity of TA1357 and of TA1414 were maintained until late logarithmic growth phase. These results indicated that the expression of GLTA was turned off by the synthetic GLTA off switch, resulting in the effective conditional disappearance of CS activity. Finally, we see the effect of switching GLTA off on growth, glucose consumption and acetate production. IPTG was added to TA1357 at various time points, that is 0, 6, 9, 12 and 15 hours. The graph A shows addition of IPTG which inhibits growth, the B graph shows glucose consumption, and the C graph represents that on later addition of IPTG there is smaller increase in concentration of acetate. And here what happened on switching GLTA off in TA1357 and TA1414 is also mentioned. These results suggested that switching GLTA off decreased the metabolic flux towards the TCA cycle, resulting in growth inhibition and increased acetate production. Here to confirm the changes in intracellular metabolites associated with switching GLTA off. Here IPTG was added and the cells were collected before and after switching. We found intracellular concentration of intermediate metabolites in the first 5 steps of glycolysis where they were not changed, and at remaining steps they got changed. We also found the intracellular citrate levels with and without switching GLTA off, which are mentioned here. And at last, coming to the results of isopropanol production, in the first figure, the switching in TA1415 was induced by the addition of 0.1 millimolar IPTG, and in the wild type strain TA1424, the isopropanol production was used as control strain. Here, when the switch was off, the cell maintenance and growth would occur, but a small amount of isopropanol would be produced. However, when the switch is turned on, the isopropanol synthesis rises along with the cell maintenance and the growth. So, this is all about the results, and now I hand over the further overall discussion of this research paper to Mrunal Ghare. Thank you.
Thank you, Akhil, Gargi, and Neha for your parts. Now, coming to the conclusion. In the bio-based production of chemical compounds, the competitive relationship between bacterial growth and or cell maintenance pathways and the desired production pathway usually results in decreased titer and yield of the target compound. In particular, carbon flux into the TCA cycle significantly competes with bio-based production pathways, which requires precursors that are important in the TCA cycle, such as acetyl coenzyme A, pyruvate, and phosphoenol pyruvate. Now, in order to improve the titer and yield of the target product, both the overexpression of the genes responsible for target compound production and the deletion of genes responsible for byproduct synthesis are required. However, deletion of these genes would decrease the growth rate and or final cell density, perhaps resulting in cell death. Therefore, these genes are rarely knocked out in metabolic engineering since the final cell density is important for the total titer and yield during fermentation. Hence, to address these issues, the researchers in this study have constructed a metabolic toggle switch in engineered E. coli as a novel conditional knockout approach for metabolic engineering and applied it to isopropanol production. As a result, carbon influx into the TCA cycle was interrupted and excess carbon flux was redirected towards the synthetic isopropanol production pathway following sufficient bacterial growth. Use of this metabolic toggle switch demonstrated a novel approach for intentionally switching intracellular metabolism as appropriate from bacterial growth phase to bioproduction phase by direct regulation of specific metabolic flux. This is a general strategy which could be applied to other bioproduction in order to enhance metabolic flux to synthetic metabolic pathways while minimizing negative effects on bacterial growth and cell maintenance. In future, additional tuning for sensitive response and optimization of the glutamine A expression level will allow further improvements in production for titer and yield. To conclude, we can say that the future aspects of this metabolic toggle switch can be that there would be no need for deletion or overexpression of the desired gene, which has multiple functions responsible for increasing the product yield. Thank you.